Hello everyone, welcome to the webinar. Today, I'll give you an introduction of our human hWGS and human hWES product pipeline. This webinar contains 4 parts. I'll start by introducing human hWGS product pipeline, followed by human hWES, then bioinformatic analysis. Finally, I'll give you 2 case studies to demonstrate how our products can benefit your research. First, let's begin with human whole genome sequencing, in short for human hWGS. Many of you may have heard of Human Genome Project. It was the most well-known exploration of human genome and has become the basement of modern human genome research. Based on that, the human hWGS is to sequence the whole human genome using high-throughput sequencing techniques. So, what can human hWGS be used in your researches? Normally, with the help of human hWGS, researchers can gain information of the genetic constitution of individuals and detect all kinds of genetic variants simply by doing one single assay. The detected genetic variants can be divided into 4 categories as shown in the figure below. They are single nucleotide variant, insert and deletion, copy number variant, and structure variant. These 4 variants are all included in our analysis progress. So, what's the highlights of our human hWGS service? First one is the sequencing scale. We deliver a high resolution and comprehensive view that covers the whole human genome. Meanwhile, our variant detection service reveals all the 4 kinds of variants in the genome, regardless of coding or non-coding regions. Besides, since human hWGS don't need us to enrich the genome, we can generate the genomic information with more uniform coverage. With all these advantages, our human hWGS service can benefit the researcher in cancer, genetic disease, human population evolution, and pharmacogenomics. In this figure, you see how our project workflow is proceeded, from sample receipt, library preparation, to the generation of the final data. Now, I'm going to introduce the pipeline step by step. Let's start from our sample guidelines. We accept a wide range of sample types, from nucleic acid to tissue samples. You can see the specific amounts required for different sample types in the table below. You can also consult our local sales for the full version of sample guidelines, which will guide you on how to extract DNA from different kinds of samples and related sample requirements. Apart from nucleic acid, we also accept tissue samples, including blood, oral swabs, saliva, cells, FFPE in slice or rolls. If your sample type is not included here, please also contact us and see what we can do. The next step is sample quality control, in short for sample QC. This step aims to test whether your sample meets our sample requirements and can proceed to library preparation. In Novogene, we have 2 methods of sample quality control: manual protection and automatic protection. Manual protection means that the project will be processed by manual protection pipeline. Well, automatic protection means processing the project with automatic pipeline. Samples will be tested in 3 criteria: integrity, purity, and concentration. In manual pipeline, we use aqueous gel electrophoresis to test the integrity and purity. And we use qubit fluorometer for concentration. Well, in automatic pipeline, we use Engine 5000 and 400 for all 3 criteria. After the sample QC, we will generate a QC report. The report contains description of your sample and final results. Figure like this will be included in the report. This figure shows what it will be like if the sample is degraded, or with RNA contamination or protein contamination. Such impurity will influence the further sequencing progress. And the table on the right shows part of the influence caused by these conditions. The sample QC report may have 2 kinds of results: pass and fail. Pass means the sample is qualified and the lab preparation can be prepared regularly. Well, in contrast, fail means your sample don't meet our sample requirements. We highly recommend you to resend the sample. Otherwise, the library could be constructed at high risk, and the sequencing quality is not guaranteed. However, if you insist, we still can prepare the library and do the sequencing. But this is with risk, and you need to take the risk. But generally, this is not recommended. If your sample passes the sample QC, we will move on to library preparation. We have 2 different modes of library preparation: normal mode and PCR-free mode. This slide shows the normal mode. In normal mode, genomic DNA was randomly shared into size of 350 BP. Then, DNA fragments were end-polished, A-tailed, and ligated with the full-length adapters of Illumina sequencing, and followed by further PCR amplification. In this slide, we can see we add the PCR step here. And in this slide is the PCR-free library. That is a bit different from the PCR mode. Is in this mode, we add adapters directly to both sides of the DNA fragments. And after this, we use the constructed library directly to the sequencing step without use PCR amplification. And finally, we generate the library like the figure below. As we can see, the library in the PCR mode is completely different from that in the normal mode. After the library is prepared, it will undergo library quality control to check if the libraries are available for sequencing. Then we will go to the sequencing step. In Novogene, we use Illumina NovaSeq 6000 platform for human hWGS sequencing. And the main sequencing strategy is paired end 150 BP. However, other sequencing strategies also available for us. Please consult our local sales for more options. And our recommended sequencing depth varies from the research field and tissue types. For genomic diseases, we recommend 30 to 50x and generate 90 to 150 G of data. Well, we recommend more than 50x for tumor tissues and 30x for adjacent normal tissues and bloods. However, the specific sequencing depth is up to you in your project. From sequencing, we can generate corresponding data. For this data, we will generate a report and deliver to you. Well, if you purchase our bioinformatic analysis service, we will analyze the data for you according to the different analysis items you order and generate a final analysis report. Details of data quality control and bioinformatic analysis will be included in our third part. So, I guess some of you may have stopped like this. I don't need to sequence the whole genome. What should I do if I want to focus more on the specific coding sequences? The best answer for this is our whole exome sequencing service. Exomes takes only 2% of the whole genome, but it is the region that encodes the protein and contains most of the genetic information. So whole exome sequencing is becoming a cost-effective alternative to hWGS, which targets only the protein coding region of the human genome responsible for a majority of known disease-related variants. In Novogene, with high sequencing depth, our service provides an accuracy detection of variants with frequencies lower than 1%. You can yield 85% of human disease mutations by only sequencing 1% of human genome, which made hWES a cost-effective method for human genome research. And this slide is our human hWES service workflow. You can see it is the same as that of human hWGS. For the sample guidelines of human hWES, similar to hWGS, we accept both DNA and tissue sample for human hWES. Please see the sample requirement of DNA in the table below. If you would like to receive our recommendation for sample extraction, sample requirement for tissues, please contact us. This page is about our human hWES library preparation workflow. We use Engineered Select Human All Exons V6 kit for exome capture. For library preparation, genomic DNA was randomly shared into short fragments with a size of 180 to 280 BP. The obtained fragments were end-repaired, A-tailed, and further ligated with Illumina adapters. The fragments with adapters were PCR amplified, size selected, and purified. Hybridization capture of libraries was then proceeded. Prepared libraries were hybridized in the buffer with built-in labeled probes, and magnetic beads with streptavidin were used to capture the exons of genes. Subsequently, non-hybridized fragments were washed out, and probes were digested. The captured libraries were enriched by PCR amplification. After library preparation is the sequencing step. Similar to human hWGS, we use Illumina NovaSeq 6000 for human hWES, and the sequencing strategy is also paired-end 150 BP. For the recommended sequencing depths, for Mendelian disorder or rare disease, we recommend over 50x depths and generate 6G of data. Well, for tumor sample, we recommend over 100x of sequencing depths. So far, we have finished the sequencing progress and generate the raw data. If you haven't ordered our bioinformatic analysis service, after the sequencing step, you will receive the raw data. However, if you order the bioinformatic analysis, we will process the data quality control and generate clean reads before data analysis. It is worth noting that only clean reads can be used in the followed analysis. So, in order to get the clean reads, we need to trim the raw data. As we can see in this figure, we remove reads contains adapter contamination. Reads with uncertain nucleotides constitute more than 10%, and reads with low quality nucleotide constitute more than 50% of either read. Low quality means reads whose base quality less than 5. We will also do statistics of the sequencing quality and generate a table as below. At Novogene, we guarantee a Q30 score of more than 85% of data of the past sample. The pie chart shows the raw data filtration result. Our report also includes evaluations for the data result, such as sequencing error rate, GC content distribution, and sequencing quality distribution as displayed in this slide. After the data quality control, clean data is used for bioinformatic analysis. Data analysis in Novogene contains standard analysis and advanced analysis. Standard analysis is the basement of the advanced one. This table shows the workflow of standard analysis for human hWGS and hWES. After data quality control, we align the clean data with corresponding reference genome and do statistics of sequencing depth and coverage. After the alignment comes the germline mutation detection. For germline mutation detection, we call the variants including SNV, indel, CNV, and SV for human hWGS, and SNV, indel, and CNV for human hWES. Then the next step is to annotate the variant codings and do statistics. However, if you provide paired tumor samples, we can offer another kind of detection named somatic mutation detection. We call somatic SNV, indel, CNV, and SV for human hWGS, and somatic SNV, indel, and CNV for human hWES. So, if you would like to call somatic variants with Novogene, please do not forget to send us normal tumor paired samples. Besides, for human hWGS, we also display the genomic variants with circles. All the subjects I mentioned above will be included in your final report. If you would like to know more about what the results are like, please contact us for the demo reports. This slide is a result generated during the sequencing alignment. The figure on the left shows the relationship between average sequencing depth and coverage in each chromosome. We also do statistics of mapping, coverage, and sequencing depth in each sample, and the result is displayed on the right. Now we come to the germline mutation detection. On the left is a detection of SNV. SNV constitute the largest class of genetic variants in genome. In Novogene, we do statistical analysis for the detected SNVs and make a summary table as shown in this slide. We also plot pie charts as below to show the number of SNVs in different kinds of genomic regions and the constitution of different types of SNVs in coding region. The results of indels are in the same mode as that of SNVs, and we can see this on the right. Apart from SNV and indel, we also offer both CNV and SV detection for human hWGS, and only CNV for human hWES. Firstly, we summarize the detected CNV and generate the table on the left. Then we plot a figure about the size of genomic regions affected by CNVs in each sample. Different colors represent different samples here. We do the same thing for SV detection and generate the table and figure like below. For human hWGS only, we can perform circles for visualizing the genetic variants of whole genome. Novogene show circles only when CNV analysis is carried out. The figure consists 7 rings from outer to inner and contains information including chrome, SNV or indel density, CNV inference. After analysis is finished, the mutations are annotated to the genome. Annotation information includes file parts, chromosomal regions, and gene structures related to this variation, database annotation, functional prediction, basic information on the variation, and gene function and pathway annotation. Here, we can see an example table of the annotation result contains the file aspect I mentioned above. After introducing the standard analysis, you may have questions like, what can I use the data in standard analysis for? Can I have a deeper sight in the analysis result? For these answers, let's introduce our advanced analysis. Please attention, results of standard is necessary for advanced analysis. In Novogene, we offer 5 advanced analysis packages, 4 for cancer research, and 1 for genetic diseases. You can decide which one to choose based on your research purpose. You may also gain inspiration based on the service category we offer. Next, I'll start from cancer research packages, then go to genetic research packages. The first cancer package is tumor progression package. where we have 3 sub-projects: intratumor heterogeneity analysis, tumor purity or probability estimation, and tumor evolution analysis. Intratumor heterogeneity analysis aims at detecting different subclones within one tumor tissue. Well, we use tumor purity or probability estimation to estimate the proportion of tumor cells and the absolute copy number of the tumor genome. If you would like to study evolutionary relationship among samples or different groups of people, then tumor evolution analysis will be suitable for you. The second package I would like to introduce is mutational profiling. This package contains 3 sub-projects: mutational spectrum and mutational signature allows us to detect the proportion and the pattern of each type of substitution across tumor samples. In order to determine the key genes and pathways critical for cancer initiation and progression, we apply significantly mutated genes and pathway analysis. If you are interested in create links between mutated genes, we have a subproject named mutation relation testing of significantly mutated genes to suit your need. In tumor researches, we have a concept named driver gene or driver mutation, which means the genomic variants that confer selectively growth advantage to tumor cells. Targeting this special mutation type, we have driver gene and mutation analysis, where we can identify driver somatic CNVs, driver mutations in non-coding regions, and driver genes based on mutation clustering bias. Besides, for the driver genes, we also conduct the pathway analysis. This is another kind of important gene known as predisposing genes, which are mutated genes that associate with the increased susceptibility to cancer. So we also include the screening of predisposing genes and prioritization of candidate genes in this package. Apart from the 3 packages above, we have independent advanced analysis. Our tumor neoantigen identification enables us to identify personalized or tumor-specific variant antigens. Well, to select human tumor mutations from xenograft, we got xenograft tumor analysis, in short for PDX. Besides, we use integration site identification to identify the integration site of transgene. We also offer fusion gene detection and CRISPR-Cas9 on and off target detecting to support your needs with different research purposes. Apart from cancer research packages, we also offer advanced analysis categories for genetic diseases and are listed below. If your project is not related to the cancer research, then it is recommended to consider the following projects. The only thing that I need to emphasize is RH analysis, de novo mutation analysis, and linkage analysis. These 3 ones, they are family-based products. If you would like to order them, please don't forget to send us samples from different family members. I guess some of you may have questions like this. What's the difference between human hWGS and human hWES in the area of advanced analysis? Please see the analysis categories are listed on the right. For this service, they are only available for human hWGS, and that is the biggest difference between them. So, in the case study, I'll show you about how we can assist you in your research using two articles powered by Novogene as examples. For this article, it is an application of human hWGS and human hWES. The author integrated human hWGS and hWES technology and demonstrated the important role of HPV integration in the cervical adenocarcinoma genomic changes, cooperating with Novogene. Cervical cancer is becoming the biggest health hazard for women groups nowadays. Among all the cervical cancers, cervical adenocarcinoma is a type of tumor that accounts for a high percentage. Previous studies have reported a high proportion of integration in cervical cancer during human HPV infection. But that leads to a question. What is the relationship between the HPV integration and cervical adenocarcinoma? This article creates this link. In this experiment, researchers use human hWGS and human hWES to sequence and compare genetic changes between a cohort of 20 patients with and without HPV integration, and called SMP, indel, CNV, and SV, which is part of our standard analysis. Furthermore, variants and amino acid alternations, driver genes, and significantly mutated genes are detected and identified by our advanced analysis. As a result, with our standard analysis, a large amount of CNV and SV and somatic mutations are identified by hWGS and hWES, respectively. And with our advanced analysis, they also identified several driver genes and significantly mutated genes. As a result, their experiment found that it is HPV integration rather than HPV infection that results in different genomic changes in cervical adenocarcinoma. For the next article, this article is an example of the application of human hWGS advanced analysis. In this study, CRISPR-Cas9 from Novogene is applied to provide genetic screens for drivers of aging and rejuvenation in old cells to identify previously unknown gene manipulations, which have the potential to provide strategies to counter the regenerative and cognitive decline with aging. Neuron protection and neuroregeneration are essential functions for animals' neuron system, and the cells responsible for these functions is named neural stem cells. But this function is always impaired by aging. Previous studies found that a large bunch of genes were observed to influence the process of aging in neural stem cells. Knockout of some of whom increased the old neural stem cells activation and leads to rejuvenation in old brains. However, so far, no systematic functional testing of genes has been done to identify the most promising rejuvenation interventions. Here in this project, neural stem cells from 6 young and 6 old mice are being tested. Researchers use CRISPR-Cas9 screening platform and a sgRNA library targeting protein coding genes and control genes to screen genes that impacting NSC activation. They also applied GO analysis to identify the functions of gene knockouts that proved to increase old neural stem cells activation and rejuvenation. Furthermore, they also used in vivo CRISPR-Cas9 screening system to study if identified gene knockouts could also play the role of restoring old neural stem cells function in vivo. So as a result, CRISPR-Cas9 platform reviewed over 300 gene knockouts that rejuvenate old neural stem cells and finally determined the specific gene that promotes neurogenesis in old individuals. And they also gained a conclusion that the process of aging in neural stem cells could be influenced by some specific genes. Knockout of some of whom increase old neural stem cells activation, which leads to rejuvenation in old brains, which provided a possible therapy for the loss of neural cells in old brains. After hearing so much of our service categories, it is important for me to make a brief introduction of our company. You are going to gain information about who we are, what we do, and why choosing Novogene. Novogene is a leading provider of genomic services and solutions with cutting-edge next-generation sequencing and bioinformatic expertise. From being formed in 2011, we are always holding the creed of advancing genomics, improving life as our top mission, and aiming at becoming the global leader of genomic services we are working at. With a corporate value of professionalism, innovation, integrity, and partnership, our skillful and highly cooperative team is always here to provide the most professional technical service and broad range of product categories to assist your researches. So far, we have built 6 labs and 16 subsidiaries around the world. Our business has spread to 70 countries and across 6 continents. Meanwhile, we have broad customer bases. Our customer covers 99% of the world's top 100 scientific research institutions and 9 of the top 10 global pharmaceutical companies. With the help of Novogene, our previous customers have published more than 950 ICI articles and have accumulated more than 7,970 of impact factors and have gained high comments from our customers. We also equip ourselves with series of cutting-edge instruments, including Novogene 6000, PacBell, nanopore, and different kinds of mass spectrometer. With this, you can support your researches with the most advanced techniques and wide ranges of choice. We are also proud of our fully automated, agile, and intelligent delivery system named Falcon. With 5 major centralized solutions and 66 outstanding devices, Falcon system can not only help you save the budget, but also shorten the project turnaround time. In order to provide you with the highest quality of data, we also created the world-class data center, which can provide you with fast, accurate, and secure data delivery. And we believe with all the advantages I mentioned above, we can give you the best experience in the scientific research. And if you are interested in our company, our service, or if you have any project that want to cooperate with Novogene, please do contact us. And if you have any kinds of questions, please also contact us by email, by phone, or any other ways. Thank you.